I don't have a choice, I only have one shot choice. Are you personally?
Because it's like a multiple choice and then like 3 answers.
I wonder if it was I had so much that I
don't think she would care.
Not necessarily, but no, literally, and I made one for them.
I made it to a different place then.
No, like aggressive.
I did die again. It's in the microbiome. It's just a
I think it's a 24% max. So well.
7-3.
Well, we shouldn't be, it should be NCW. What? In finals. Yes. In the finals.
Yeah.
All right, let's get started. Good morning. Okay. Briley. Brianna. Drew. Bria. Henry. Anna. Here. Nick.
Here.
Abby. Hannah.
Here.
Rosa.
Here.
Matt.
Here.
Jillian. Mia. Annie. Brianna H. Brandon.
Here.
Sadie. Jessica. Jesse. William. Marissa. Anna. Sophia. Aubrey. Matthias. Colin. Braden. Olivia.
Me too.
Amber. Oh, hi. Hi. No worries. Go back. Katie. Kennedy. Ben. Guys, can you quiet down a little bit so I can hear people, please? Kendall? Here. Emily? Here. Lauren? Here. Jennifer? Here. Isabel? Here. All right, cool. All right, so today we're gonna talk about the human microbiome. We're gonna kind of put things into the context of just different types of microbiomes. So we'll talk about the gut, we'll talk about the skin, we'll talk about the vaginal microbiome, we'll talk about all the different microbiomes, the gut-brain axis over the next couple lectures. So some of this is introduced in chapter 23 in your book, but the rest of it is from my knowledge. So the human microbiome, we've kind of talked a little bit about this before, but it's the indigenous microbial communities of the human body. So indigenous means they're native, they're there, they don't cause harm, and so they make up the normal microbiota. Once again, microbiome and microbiota have some different definitions. The microbiome, remember, is all the microorganisms in or on the host, as well as the genetic material, whereas the microbiota is just the micropopulation in a specific ecosystem. So we can distinguish between the gut, the skin, etc. And so if you notice over here at on the top, let me turn this light off. In this image up here, you'll notice, even your hair has a microbiome, your nose, skin, vagina, colon, stomach, esophagus, and the oral cavity. If you look at these colors, they indicate different types of bacteria. You'll notice that all these different sites have different proportions of the colors. So each body site is colonized predominantly by only certain bacterial groups. There's a lot of differences. And so if we zoom in on the lungs, gastric, oral, and nasal cavities, you can see this is another image kind of showing the same thing, where there's very different bacterial communities that inhabit these different sites. And so there's definitely a reason for that, and we'll talk about some of those things. So the microbiota exists on all surfaces. They can be external or internal. So internal could be your organs, external could be your skin, your hair. And so there are significant positive effects of symbiotic relationships that we talked about between the different microbes. They help also promote well-regulated immune responses by your immune system. We know that microbiology and immunology are very tightly linked, and so the microbes can help promote good health. And like we also talked about, they can compete for potential pathogens, or with potential pathogens for space and resources. So what did we call that? Shout it out. You should know this. Colonization resistance, yes. So you guys should know this. It's going to come up a lot. So I want to start introducing you guys to some types of figures and try to break down how we would read different figures. So I'll walk you through this. This is a PCA plot, or a principal coordinate analysis. Basically what the X and the Y axis are these principal coordinates. So basically what they're looking here is what these different microbiomes, how similar or how different they are from each other. So they sampled different people's these different microbiotas, and so the PC1 and PC2 are arbitrary. So basically they take all of the variables that make these microbiomes different. There's so many different factors, and those factors are put on a 2D scale. So if oral cavity, so basically all of the orange, the microbes from different people in the oral cavity are clustered more closely together, that means that they are more similar. So the way to read this is the more close together two things are, the more similar they are. The more further apart they are on the scale, the more different those communities of microbes are. Does that make sense? So if I were to ask you, is the oral cavity more closely similar to the skin or the gastrointestinal tract? What would you say? The skin, right? Because there's a lot more overlap between the orange and the green. Great. So you guys know how to read this. And so, yeah, so this is different body sites in 300 healthy humans, just looking at the microbiota. So the key here is that this shows clusters of samples based on their similarity, and groups that are more similar to each other will have higher overlap on a PCA plot. And so the key takeaway here is that the composition and the diversity, all of those things, there's so many ways to measure composition and diversity, and that's all kind of accumulated into this plot. So we can say by looking at this that the composition and diversity of the human microbiome is strongly determined by habitat. So although I told you that based on this is another way to show it. And so this is where you get into different research methods that help you come to these conclusions. So we'll talk about different ways to basically hone in on the same point. Any questions so far? Okay. So this plot is showing the relative abundance, so how abundant these different phyla are, phyla of bacteria, in all of these different areas. So you have your nostril, RC is a retroauricular crease. It's like the crease behind your ear. Apparently that has its own microbiome. The buccal mucosa, so your inside of your cheek, the teeth, supragingival plaque, tongue, stool, and cervix in women. So what they're looking at is, you know, what microbes inhabit these different phyla, and the thing that's missing here is a scale that goes from 0% to 100% at the top. So obviously there's going to be 100% of microbes, and so you'll see here that in the nose you have a lot more red, and so the red is the actinobacteria. So you could say that maybe, like, I don't know, if you took an average of all of these, maybe like 75% of the total abundance of the phyla in the nostril are actinobacteria. So just another way to kind of show that there are differences among the different sites. Does that make sense so far? Okay. So this is looking at who's there. We're just looking at phyla. But remember, these bacteria all have different functions. So if you look at the metabolic pathways among these microbes, they actually are pretty similar. So although that the microbes in these areas are very different, they still maintain a healthy microbiota by performing certain functions. So these functions are over here. So say ATP synthesis, it's really important. It's making energy. So although, you know, there are probably certain microbes that do better at making ATP synthesis in the nostril, there are very different microbes that can have the same metabolic function, but in a different site. So although the carriage of microbial taxa varies between sites, the metabolic pathways remain stable in a healthy population. So the end goal is kind of the same. Does that make sense? Any questions so far? You know, I'm just dumping like a lot of information and scary figures at you, but when you are reading a figure like this, I always say first, you know, just look at what the y and the x-axis are telling you, and then kind of build your way up from there. That's always a good start. Okay. So what do you think, you know, from the moment we're born until our lives today, what has determined the composition of the microbiomes that we have over time? You know, so if you look at it over time, from when you're literally in the womb till adulthood, and then even like geriatric age, they definitely vary over time. So what are some factors that could influence our microbiome? Let's go through some of these together. We can, just based on what you guys think. Yeah. Genetics, yes. Yeah.
Nutrition and lifestyles.
Diet, lifestyle, yeah. Yeah. Illness, if you're sick, for sure. And then if you're sick, say with an infection, what are you given? Antibiotics. Antibiotic exposure will influence your microbiome over time. What else?
I guess just overall immune system health.
Immune health, yeah. How competent your immune system is. What else? Right now we're all sitting in this classroom, and so we're all, breathing the same air, and so we're definitely exchanging microbes. So what kinds of environments can shape? What? Someone say something? So what kind of environments could affect your microbiome? Yeah.
It'd be like, kind of like, what kind of, I remember I learned what the word was, but like, what, like temperatures like you live in, like if it's drier compared to like if it's
Yeah,
wet.
absolutely. Different environments, different seasons, different temperatures, 100% that'll influence your microbiome. Yeah.
Whether you grew up in like a densely populated area or like a more rural area.
Yeah, 100%. Your where you grew up definitely influences it more long term. If you're in an area that's very urban and there's a lot of pollution and factories versus if you're in a very rural area, if you lived on a farm with animals. What about if you have pets? You guys know that there's a lot of studies that have shown that if children grow up with pets, they are less susceptible to getting allergies to pets because you're priming the immune system from a young age with exposure to animals, and so they're less likely to develop allergies. There's so many things. What about, think of the moment you're born. What are some differences in how people can be born? Yeah.
C-section.
C-section versus vaginal birth. That is a huge one. We're going to talk a lot about that today. What else? What happens to, what does the baby need from mom when baby's born? Yeah. Yeah, breastfed versus formula fed. There's so many different things. So what about, a lot of you guys are very active, a lot of you guys are athletes. Do you guys think that might influence your microbiome? Yeah, absolutely. Exercise. A couple of students in my advanced micro class did a presentation on the effect of basically runners and endurance, training on the microbiome. So there's a lot of papers out on that. Let's see, what else? So we'll talk about a lot of these in more detail, but these are some of them. And so, yeah, there's so many things. Even your sleep will affect your microbiome. And so if you notice over here in yellow and this arrow, this is actually measuring microbiome diversity and stability over time. So when you're just born, it's not as diverse, you're still developing it, and it's not as stable. And then as you grow up, it becomes more and more stable. Childhood through adulthood is probably when it's most diverse and stable if you're a healthy individual. And then in the geriatric phases, you kind of become a kid again, it starts to decline again. So, it's important to take care of our microbiomes now so that later on, it definitely helps long term. But one of the things that is very interesting is that there's critical microbial exposure that happens at birth. So the first thing is the maternal microbiota, the mom's microbiota that mom will transfer to the baby, include, going through the vaginal canal. The baby is actually receiving a lot of important microbes from mom that help prime that baby's immune system. And so even, while the baby is in the womb, it's actually building that microbiome from the mom's intestinal tract. And so the next one is breast milk. Obviously, there are so many nutrients and positive, microbes and things in breast milk. And so, not to say formula-fed babies are never going to be, healthy or, never going to be able to have a stable and diverse microbiome, but there's definitely differences when you're born and you're being, breastfed versus formula-fed. There is a lot of research done that infants that were both formula-fed and born via C-section have more susceptibility to developing allergies, asthma. I make a clear case of that. I was not breastfed, I was born C-section, and I have allergies, asthma, and immunocompromised. I have all the things, but we're still fine. But yeah, so that definitely makes a difference. I only lost my spleen at 19, and we're still living. Yeah, so. And then if you guys have heard of skin-to-skin contact, that is so important. Your skin microbiota, literally that contact helps the baby build a lot of those microbes. Any questions so far? And then, as baby is starting to grow, it starts to seed its own early life microbiota. So, this is when even when it's still a fetus, there are other ways to develop certain microbes. And so metabolites are certain compounds that are produced by the microbiota that are good for you. And so, even through the placenta, there's signals that are sent to the baby. And so obviously we talked about breast milk and the nutrients in there. There's antimicrobial peptides in there. And so, vertical transmission through the vaginal tract. And then, to seed that early life microbiota with really important microbes, that definitely leaves a lasting imprint on the biology of that child as it grows. And so this helps with really, a strong immune system development in the early life of the child, as well as neurodevelopment. So like I said, susceptibility to childhood illnesses include infections, asthma, obesity, neurodevelopmental disorders. So there's really a lot that goes into this beyond just what the baby, the microbes the baby gets, but also, longer term, there's longer term consequences for sure. Okay, any questions so far? Okay. So now I'm going to show you what I just told you. And so this is looking at, the effect of birth mode and feeding mode on the microbiome composition. So if you just kind of ignore B, C, D, E, and F, just look at A. These colors represent the different types of microbes, and it's really nice because they literally give you everything from like the phyla all the way down to the genus. So even though there's like a lot of different Firmicutes, they even went further in and figured out what species made up each. So, you know, you can see that very, very early on in life, you have a lot of proteobacteria. And so, you know, as you grow, you start to build, and then it becomes more stable. You know, after maybe like 10, 11 months, you start to see less and less change. So this is all the subjects in the first 2 years kind of put together. But these subjects actually have differences. So then they split them into these groups where they were looking at those that were breastfed and born vaginally, which is really the best case scenario. If they were vaginally born and formula fed, breastfed, born C-section, and formula fed and C-section babies down here. So these, just to also note, these babies did not have exposure to antibiotics during these months of life because that would significantly skew the results. So that's something that is, you know, supposed to be controlled for. So yeah, you can definitely see, you know, best case scenario, this is essentially what it would look like. This is from 0 to 6 months of life. And so you can see already that formula-fed infants at the very start, they don't have a lot of those, you know, bacteroidetes, which are very important in producing short-chain fatty acids. We'll talk about that as well. You know, formula-fed have a lot more of this light blue. You know, same with C-section. And then look at all that orange that pops up in the C-section of formula fed. So that just, you know, tells you that there are differences, and the microbiome is shaped by birth mode and by feeding mode. Any questions? Okay, so to sort of kind of now put this into some more context, the maternal microbiota and the immune system adapt to different stages of pregnancy as well. And so here, the maternal microbiota basically produces certain, like I mentioned earlier, metabolites. Those metabolites can be, you know, fatty acids that are really important. They have positive effects. They reduce inflammation and things like that. And so, you know, there's a list of them here. That doesn't matter too much for you guys to know at this point, but there is placental, you know, metabolites that, you know, get transferred to the baby as well. So, and that helps prime the immune system, because if you see when those metabolites are there, you start to get, you know, recruitment of these different immune cells. That just helps, you know, make sure that there are immune cells there to protect the baby. And so, you know, once again, during vaginal birth, the maternal gut and the vaginal microbes represent the main source of microbes for neonatal microbiome assembly. So this is when the baby is in the womb still. And so breast milk is the main postnatal source. So, you know, vaginal microbiome and the maternal gut, the mom's gut, are the prenatal source of microbes. Breast milk is the postnatal source of microbes. And so also helping develop the immune system. And the composition of the breast milk is complex and unique to each individual. There's studies that have shown that mom's breast milk develops based on what the baby really needs. And so just so, you know, I'm sure maybe you guys know this or not, but during breastfeeding, it's a positive feedback loop. So the infant has to basically suck in order for the mom to produce milk. And so there's a reverse flow of milk from the infant back into the breast, which actually can serve as information to tailor the milk to the infant's needs, which I think is really, really cool. Now, kind of moving on from the maternal and child microbiota, environments are sites of continuous microbial exchange. So here you can see that there's human shedding, pet shedding, outdoor air, soil, indoor mold growth. All of that's continually, you know, being exchanged through the air. That's why when we work with microbes in the lab, and especially with the fungi that have spores, I tell you guys to make sure the lid is on when you're not actually looking at something, and that's because there's things that are constantly being exchanged in the air. So you want to be mindful of that, even the things you can't see with the human eye. Environmental signals, you know, could be pollution. It could also be introducing something like antibiotics. And so diet obviously is also a big one, and we'll talk about that. Dietary fiber is known to be very good for you. It helps to improve your microbial diversity and metabolism. And the gut microbiome signaling, basically producing these metabolites and these things, allow for communication with organs and tissues in the host. So it affects everything from, you know, your fat tissue, your liver, brain, your heart, your lung. Everything is influenced by your gut microbiome. Now, the gut microbiome and diet, it's a big one that's been very, very widely studied. So basically, you know, the healthier you eat, if you eat more of a Mediterranean diet, you have better microbiota symbiosis. So symbiosis is these positive interactions that occur between microbes. Dysbiosis means that there's an imbalance in the microbial communities. It's dysbiotic, so you'll hear that term a lot. It's been shown that a high fiber diet helps to, you know, aid in producing, you know, allowing these microbes to grow and reproduce and take over the habitat and exert colonization resistance on pathogens. So if you have a high fermented food diet, you know, that results in you have, you know, microbes that aren't necessarily good, and they end up having more negative effects on health. One of the things I want to focus on is basically the microbiota produces metabolites, and short chain fatty acids are an important one that we'll talk about. But just to kind of help you guys understand more like why dietary fiber is important, dietary fibers, basically starch is a really, really important one. Basically, starch is a polymer of glucose. And so it's made by plants to store energy. And there's different types of starch. There's accessible starch and there's resistant starch. Accessible starch is starch that is able to be cleaved by human amylases. Amylases are enzymes in our saliva and in our body that can break down this accessible starch. So it's something we eat and we're able to break it down. But there's another form of starch, which is called resistant starch. It's very crystalline, and our enzymes cannot degrade this. Rather, bacteria have enzymes that can degrade this resistant starch. The resistant starch is actually very good for us to eat because we don't break it down, but it's food for the good bacteria in our gut, so they break it down. And when they break it down, they produce these short-chain fatty acids. Does that make sense? So this process is called colonic fermentation. Basically, these microbes, these bacteria, are using the resistant starch, and this is called fermentation, one form of it. And this results in short-chain fatty acid production. So I'll walk you through this. Here we have epithelial layers, the epithelial cells, and then you have this mucus layer on top that helps make sure that, it's protecting the intestinal tract. And then on top up here in the lumen, say we eat a plant particle. So this is the dietary fiber. And so there are different degraders, basically different microbes that are able to break down this plant particle. And so there's primary degraders, there's secondary degraders, but what matters is that through this process of degrading these fibers, that produces short-chain fatty acids. And so the four main ones that you guys have to know are butyrate, propionate, lactate, and acetate. Butyrate is the most widely studied short-chain fatty acid, and its effects are very positive on the intestinal tract. These are known to help improve, make sure that the mucus layer stays thick. They help reduce inflammation. So producing these short-chain fatty acids is really good. And the microbes do it. So the more we feed the microbes with these fibers and resistant starches, the healthier our gut will get, essentially. Questions? Okay, so another way to, put this together, you have short-chain fatty acid production, and these have important functions on the epithelium and on the immune system. So it helps increase epithelium health and function and decrease inflammation and the immune cells that make inflammation in the body. So the production of butyrate is very important in the function of the gut microbiome. So butyrate is the conjugate base of butyric acid, so you can hear it either way. I usually say butyrate, an end product of fermentation by microbes in the colon or the large intestine. That's why I called it colonic fermentation, because it's in the colon. There are some beneficial effects of butyrate. It's the preferred energy source for mitochondria in the cell lining in the colon. Helps decrease inflammation, decreases the likelihood of cancer, reduces incidence and severity of graft versus host disease. It's basically when your body starts to attack itself, it recognizes self as foreign, and it's an autoimmune disease. So butyrate has a lot of good function. And when I was a freshman in college, I took a general biology lab that was a little bit different. Basically the professor was trying to study the effect of supplementing diet with resistant starch on butyrate production. So I took this class and we were able to be basically in the study. So we were given either a placebo or resistant starch supplementation. It was a powder that we would put in our water and drink it over time. And so basically so I am actually one of these dots. And so we looked at butyrate levels. How do you think we looked at butyrate levels? We had to collect our own feces. So yeah, that was something I had to do. They gave us a kit, and we basically had to put this piece of paper in the toilet and collect a little sample of the feces. They gave us everything, gloves, and then you like put it into the solution and mix it, and then we drop it off, and then the scientists in the background would go and, you know, extract the butyrate levels from it and test it over time. And so that's why I was so interested in microbiome research, because that was like my first experience with it. I was a freshman, and then the next year they asked me to come back and help teach the class as like a TA, and that's how I got into microbiome research and teaching. But you know, this showed, and this paper was published in 2016, that before resistant starch in in blue, you know, levels were lower for most of the individuals. Some of them there was no change, some of them there was really a decrease, so it's not the same for everyone. And you know, if we go back to the things that can influence our microbiota, you know, all of us who tried to, all of the students in the class that did this, we tried to control for differences, but there were so many differences between us, you know. Some of us were not, you know, we're born differently, C-section versus vaginally, breastfed versus formula-fed. Where you lived was a big thing. There was a study on the microbiomes in different dorms, and there was differences. So there's so many differences, even in the way we grew up, our genetic background, our cultural background. So there's so many things to consider, but you know, ultimately, this result was statistically significant. So a P that's less than 0.05 is statistically significant. So we had it at 0.01, so it showed that resistant starch supplementation increases butyrate production. So that was definitely really interesting exposure to research, but you know, so any questions on that, I guess? Does that make sense with the resistant starch supplementation? Okay, so like I said, the microbiome basically undergoes a lot of different mechanisms to prevent the invasion of foreign populations into this established community. And so these are the indigenous populations, remember? And so if you introduced antibiotics to this established community, what mechanism is disrupted? So I'm telling you that these microbes prevent the invasion of foreign populations, and then we introduce antibiotics. What are we doing with antibiotics? We're removing the majority of these microbes. So what does that do? It lets the invaders come in, exactly. So it disrupts colonization resistance. I told you guys, this is gonna come up a lot. When in doubt, colonization resistance. This is why, you know, antibiotic use really can influence the, you know, development of secondary consequences, secondary infections. One of those is the fungus, Candida albicans, that I studied for my PhD. And basically this is also a member of the indigenous populations, but it's kept at very low levels by these microbes. They keep it at low levels, it doesn't do any harm. If you introduce antibiotics, antibiotics kill bacteria, not the Candida albicans, because that's a fungus. So it's a yeast. So once you introduce antibiotics, it depletes the bacterial microbiota, and then that fungus there has a lot more space and a lot more nutrients. And so it can overgrow and take over. So that's why in a lot of females, if you get a UTI, a urinary tract infection, and then you take antibiotics for it, a lot of times you'll get a yeast infection after, because taking antibiotics disrupts the bacterial microbiota and allows that yeast to overgrow. So a lot of women struggle with this. And so it's really important, you know, to understand why antibiotics are great, but also can be harmful. Okay, so I will stop here and we'll go through the rest of this on Friday.
Not even exercise-induced asthma, just like general asthma. So if like, do you remember the wildfires from Canada? A few, like it was it last summer or some? But I remember my friend would go outside, and be like, oh, it's really smoky. I'd like walk outside and just be like, because like it just gets triggered so easily. Because my mom, she actually has a hip problem, so she had to have the C-section because she couldn't like give birth properly. Both me and my sister were both C-sections because her hip is like messed up, so that's why she couldn't like, you know, I don't know everything about it, but like contraction and stuff.
Wow, yeah.
Asthma.
Yeah, that's how it goes.